Welcome to my channel. So in today's video we are going to discuss about restriction endonuclease. So restriction endonuclease or restriction enzyme, or also this is called as molecular scissor. So these are bacterial enzymes. That is, they are produced only in bacteria. And the special feature for this particular enzyme is that they can cut DNA molecules internally at specific base pair sequences. Specific base pair sequences are called as recognition site. So generally every restriction endonuclease that has got a specific recognition site. So wherever this recognition site is present in the genome, this restriction endonuclease will go and bind there, and then subsequently they will cleave DNA molecule at that particular point. Now discovery of this particular enzyme, restriction endonuclease, this has made molecular cloning feasible. So as you know, what is molecular cloning? Very basic concept is, suppose there is a gene present in one species and you want to clone that into another species which doesn't have that gene. So that is the very, in layman language, what this molecular cloning is. So the basic concept or basic thing that is needed for this molecular cloning is to fragmentize or break down this whole DNA molecule, whole genomic DNA, into very small pieces, and then two of such pieces from different organisms, they can be joined together. But prior to this discovery of restriction endonuclease, the process that were used are small bore needles or sonicators. These methods, they do not have the specificity. That is, they cannot cut the DNA at one particular position every time. So whenever you are using this sonication or small bore process, every time they will fragmentize a new section of the genome. So neither the total number of fragments will be same, nor the size of each of these fragments will be same. So this is not specific, and this cannot be used for molecular cloning, which basically got possible only after the discovery of restriction endonuclease. And the credit for discovering this restriction endonuclease enzyme, this goes to these two gentlemen on the left, Dr. Werner Arber and Hamilton O. Smith. And for this feat of theirs, they received the Nobel Prize in 1978. Okay. Now, these enzymes, these are formally designated as type 2 restriction endonuclease. But apart from this, there are a few other types of restriction endonuclease as well, like type 1, type 3, type 4, etc. But what makes type 2 superior over the rest of the other types is their specificity. Now let's see how does this restriction endonuclease work. Okay. So suppose this is a DNA molecule. So what the enzyme will do is they will scan through the entire length of the DNA molecule, and wherever they will find their recognition site, they will come and bind there. So suppose this is the recognition site, this is GGATCC, and on the other hand CCTAGG. So let's assume this is the recognition site for restriction endonuclease X. So this restriction endonuclease X will scan through the whole DNA sequence, and wherever they will find their recognition site, they will come and bind on it, and then they will cleave. So this cleaving is also very specific, that is, they will cleave in between any two specific nucleotides. So here in this example, this has been pointed out using or shown using this arrow, where their cleaving position is, where they will cleave the DNA molecule. So after at the end of this restriction endonuclease digestion, these are the fragments of DNA that we are going to get. Now coming to the nomenclature of this enzyme. So suppose this one, EcoR1, how the naming, this EcoR1, how the name is being given. Okay, so this namings, the naming of these enzymes, they follow a very strict nomenclature. That is, the first letter here in capital, this is designated the genus of the species from which the restriction endonuclease was extracted. The next two letters, this one in case CO, these are the first two letters of the species from which they were extracted. Like for example in EcoR1, this was extracted from Escherichia coli bacteria. Now that's why E stands for Escherichia, the first genus, capitalized letter, and the second one, CO, stands for coli. Okay. Now the fourth one, this stands for the strain name or the serotype of the source bacterium from which the restriction endonuclease was extracted. So in this case, this was extracted from the R strain of Escherichia coli. and in this case in heme3 the D designates the serotype of the bacterium from which this was extracted. Okay. And now coming to the last, that is the number 1, 3. So this is the number of restriction endonuclease that has been extracted from this particular strain of this bacteria. So suppose if the scientists now come up with, or they are being able to extract another restriction endonuclease from the R strain of the Escherichia coli, then that will be named as EcoR2. So that is how the nomenclature of this enzyme goes. Now what I have done is I have added a problem regarding this, that is what is the type of what is the name of the restriction endonuclease, which is the 4th restriction endonuclease isolated and characterized from the T strain of Helicobacter pylori. So now if you can solve this, then write the answer in the comment section. Anyway, so let's move ahead. So next we will see how the restriction endonuclease cleave the DNA molecule. So the restriction endonuclease, they can cleave the DNA molecule in 2 types. One is the staggered cleavage, which is also called as sticky end cleavage, and the other one is called as the blunt end cleavage. And the restriction endonuclease can be classified based on these characteristics. So the ones which produce the staggered end, they are called as staggered end or sticky end producing restriction endonuclease, and the other one is called as blunt end producing restriction endonuclease. So let's see how this works. So this area, this highlighted area, this is the recombination site, and these are the areas where, or these are the nucleotides rather, where the restriction endonuclease will come and cleave. So after this digestion is being over, then these are the 2 fragments that you are left with. Now the important thing here is that you can see there are few unbound nucleotides in this fragment, also in this strand, and in this fragment in this strand. Now this is called as the sticky end or the protruding end that is being produced by this kind of digestion. So this will be more clear in this particular explanation. So this was the sequence. Obviously it is a long sequence. I have only taken a small part of it. So this DNA molecule, if this is the restriction digested using an staggered end or blunt end producing restriction endonuclease, then these are the 2 fragments that are going to get produced, given that one was cut here and another one was cut here. Now, and these are the protruding ends or the sticky ends that I was talking about. Now, how does this restriction endonuclease cleave the DNA? Or where exactly does they cleave this DNA molecule? Now here this is one strand of DNA, and on opposite to that another strand of DNA is being synthesized. Now basically what happens is, suppose if A is there, then the incoming deoxyribonucleotide that would having T will be selected, as because A and T, adenine and thymine, they are bound with each other, and C and G, cytosine and guanine, are bound with each other. But the problem here is that this DNA, this hydrogen bonds here, hydrogen bonds is relatively a weaker bond. I have created a separate video on the structure of DNA, so the link will be given in the I button. So if you are interested, you can have a look at it. Anyway, coming back to this thing, so this DNA molecule, this is interbonded and they are constantly under immense amount of pressure. And this hydrogen bond, being the weaker bond relatively, this cannot withstand this kind of a pressure for a long period of time. So that's why the bond which gives rigidity or stability to the DNA molecule, that is the phosphodiester bond. And the phosphodiester bond is formed between the 3' hydroxyl group of one nucleotide and the 5' phosphate group of another nucleotide. So between this OH group and phosphate group, this is the phosphodiester bond that is being formed, and this is which gives rigidity to the DNA molecule. And restriction endonuclease cleaves this particular phosphodiester bond. Now coming back to this discussion, this is the another kind of cleavage produced by restriction endonuclease, which is called as blunt end producing restriction endonucleases. So here this is the recombination site, and these are the 2 sites where the cleavage is taking place, and after the digestion is over, then these are the 2 fragments that are being generated. So let's see here. So this was the sequence, and from this, these are the two segments that are being generated, and as you can see, there is no protruding end or sticky end present in this. So now let's see what is the role of this restriction endonuclease in recombinant DNA technology or gene cloning. Now let's see, these are two different DNA molecules, and as each of them, they have got a recombination site for one particular restriction endonuclease X. But if you see carefully, the sequence of the two more DNA molecules are entirely different from each other. In the next step, we digest both this DNA molecule with, so let's say we take both of this DNA molecule in the same Eppendorf tube, and then we allow them to be digested by a particular restriction endonuclease X. So at the end of this digestion, these are the two fragments that are going to get generated from this DNA molecule, and these are the two fragments that are going to get generated from this DNA molecule. Now here, if you see carefully, although the sequence is different from these two DNA molecules, but because their recognition site is same, which was present here, which was being cleaved by the same restriction endonuclease, so this protruding end or the sticky end that are being produced from each of these fragments are actually the same. If you can see here, G, C, T, A, G, here G, A, T, C, and G, here also G, C, T, A, G, exactly same, G, A, T, C, G, G, A, T, C, G, exactly same. So now this one and this one are complementary, but at the same time, this one and this one is also complementary. Same way, this one and this one is also complementary. So after this digestion is over, if you allow them to rejoin, then this fragment can join with this one, but at the same time this can join with this one as well. Let's see. So suppose from this fragment, these two join back together like this, or this one and these two can join among each other. Likewise, from here also, this can join back like this, or this one and this one also can join back together. So what it does, it creates new sequence of DNA, which is actually needed for the gene cloning method, or which is needed for DNA cloning. So in DNA cloning, you have to add the plasmid DNA or the vector DNA with the gene of interest. So that's why restriction endonuclease is being used. So now let's assume that this one is the gene of interest, this one is the vector DNA, both of them being digested by the same restriction endonuclease, and then one segment of this joins with another segment of this restriction endonuclease, and thereby you can join the two different DNA molecules together. So that is how restriction endonuclease is important or being applied in case of recombinant DNA technology. And as a matter of fact, among these two, that is a blunt end producing and the staggered end producing, the staggered end one or the sticky end producing restriction endonuclease, that is preferred in recombinant DNA technology. Okay, now the next question that should come to our mind is that if bacteria is producing this restriction endonuclease, and what if the recognition site for a restriction endonuclease produced in a bacteria is present in the same bacteria, right? Then hypothetically, this restriction endonuclease should go and cleave their own genome, and ultimately that will become detrimental for this organism. So what happens in reality is that this particular restriction endonuclease, this is our defense mechanism of the bacteria against the invading bacteriophages. Bacteriophages are viruses which invade or attack bacteria. Now this restriction endonuclease, this works as a defense mechanism. So whenever the bacteriophages attack this bacteria, so this restriction endonuclease go and cleave the genome of those bacteriophages. Now for their own genome, they have developed mechanism to protect their genome from the deleterious effect of restriction endonuclease. The most common strategy is methylation of the cysteine residue that is present within a recognition site. Now let's see the previous one. here this is the recognition site. This is the recognition site, and within this is the cytosine. Now let's assume this is DNA sequence of that particular bacteria. Now what they will do is they will methylate, that is, that will add a methyl group to the cysteine nucleotide, as a result of which the conformation or the structure of cysteine will change. So now the restriction endonuclease won't be able to come and bind to it and subsequently cleave to it. So that's how they protect their own genome from this restriction endonuclease. Okay, and these are the example of few restriction endonucleases, the most common ones. These are the recognition sites, and this arrow shows at what position does they cause the cleave, and this is what type of end or what type of fragment does it produce, whether it is blunt end or whether it produces a staggered end, that. Okay. Now there is another subgroup within this type 2 restriction endonuclease which is called as type 2S restriction endonuclease. What is the specialty about this is that they will go and bind to the specific recognition site, but they won't cleave within this recognition site. Now if you see the previous one, all of this restriction endonuclease, they bind to this recognition site and cleave within this recognition site. But the uniqueness with this type 2S restriction endonuclease is that they will bind to this recognition site, but they won't cleave here. They will cleave some nucleotides upstream or downstream. So what do you mean by upstream and downstream? Whenever you are having a DNA molecule 5' to 3', from any given position, if you go towards the 3', that is regarded as downstream. If you go towards 5', that is regarded as upstream. So they will either go upstream and downstream and then cleave, and how many nucleotides they will move, that is very specific for each restriction endonuclease. Like for example, FOC1, this is a restriction endonuclease. This is the recognition site for them, this GGATG and CCTSC on the other strand. It cuts 9 nucleotide downstream for the upper strand and 13 nucleotide downstream on the other strand. This is one strand 9 nucleotide, another strand 13 nucleotide. So how do you write it? How do you represent it? Either you have to write this sequence or recognition site and then how many nucleotides are there. So this N stands for nucleotide. On one strand 9 nucleotide, another strand 13 nucleotide. In another representation way where you don't write so many n's, just you write n and then write the total number of times it is being repeated. So this is 9 and 13. And in another way, here you just write the nucleotide strand of the upper strand and then you write 9 by 13. The 13 goes for the other strand. So this is how this thing is represented. And so suppose this is the DNA molecule and this is the recognition site, then this restriction endonuclease will come and bind here, but they will cleave at this position in the upper strand and in this position in the bottom strand. So this is how it goes. And these are the example of few such type 2S restriction endonucleases. As you can see, all of them they will actually go and cleave some nucleotides downstream or upstream. There are some restriction endonucleases, although they are being collected or extracted from different organisms, but they cleave or they have the same recognition site, and within that recognition site they cleave different nucleotides. That is the uniqueness. So these are restriction endonucleases which are extracted or collected from different organisms, but they have the same recognition site and within that they cut different nucleotides. Okay, so in this case, suppose 4 restriction endonucleases have been discovered which has the same recognition site. So in that case, what will happen? The first restriction endonuclease that was discovered, that will be called as a prototype, and the next ones they will be called as isoschizomers. Okay, and the example XHO1 and PAER71, these are the restriction endonucleases collected from different organisms. Okay, but they have the same recognition sequence, and another uniqueness is that they also have same cleavage locations as well. Now isoschizomers that cleave at different positions within the same recognition site, these are called as Neoschizomers. So this is an example. So for all of these 4 restriction endonucleases, they share the recognition site, this is the recognition site, and within them they have different cleavage sites. This kind of isoschizomers that cleave at different nucleotides at different position within the same recognition site, they are called as Neoschizomers. So what are the terms? First one is isoschizomers, and the isoschizomers which cleave at different position within the same recognition site, they are called as Neoschizomers. There is another type here. These are the restriction endonuclease. They have different recognition sites, but they produce the same nucleotide extension. That is, suppose for example BAM-H1 SAW3A1. So both of them, they have different recognition sites, but after the cleavage, the extension or nucleotide extension that are being produced, that will be exactly same for these two enzymes. So with that, we have come to an end to this video. Hopefully you have liked this video. If this video has helped you, then for channel promotion, kindly like and subscribe to this channel and also share it so that others can also have a look at it. Thank you.